Hello, good afternoon, everyone, and thank you for kind introduction. My topic for today is some of the challenges which we encounter when we are validating the molecular tests, especially for the global clinical trials. And some of the topics which I will be covering this afternoon is the regulatory compliance. It could be like CAPTIA, and then there's new regulations which came in very recently, like in May of last year, IVDR regulation in Europe. And we all know like the how difficult to get the validation materials, and especially some of the rare diseases, getting the positive samples. So some of those with talk, and then in the clinical trial, the other challenge which we encounter is um, asking for like different sample matrices. Like we might have the FDA approved test for one sample matrix, but then the request comes from the clients or the pharmaceutical company. Different matrices depending on the trial. So CMV as an example. So I will tell you how we do. And also always the challenge how soon this needs to be completed. The typical time we take is anywhere between 2 to 4 months, and sometimes even faster, depending on the trial need. And then once we take all this into consideration, so we want to develop really highly easily validated test, and then maintaining the quality over a period of trial, which may run up to 4 or 5 years, and thousands of samples in multiple locations and all of that. So how we make sure that goes through smoothly. And so before going into these are some of the experiences we gain, or we're working through when we need to validate these tests. And I'm going to share some of the experiences which we do it in our institute. So and PPD is a global CRO. We do support the clinical trials for all the phases, phase 1 to 4, and we have several labs located all over the world. And then the ones which are in purple, there are 4 of them, capital labs. 2 are in the Asia Pacific region, one in Singapore and 1 in China. And we also have a lab in Brussels, Belgium, in addition to having a lab in US, in the state of Kentucky. And then in addition to these central labs, then we also have the biomarker labs, and then the vaccine labs, and also the GMP labs. And here, this slide kind of represents the involvement of these different labs and on the pages of the trial. The central labs, the one which I'm the director, one of the directors, is mainly involved in the clinical development, that is from phase 1, phase 2, phase 3, and then also to the post-approval, a regulatory approval. And then our bioanalytical biomarker labs, so they cover the entire spectrum of the trial, and then the GMP and vaccine labs are equally separate in those studies. Again, just to give it the central lab services, so we do the testing to include or exclude the subject, or and so that could be initial the basic chemistry and urine analysis and hematology, and threats of abuse, and some of the infectious disease biomarkers, and any tests which are appropriate based on the trial. Then we also test some of the safety and the efficacy testing. Again, that could be depending on the trial, but all of them have the routine chemistry, the hematology assays, in addition to some of the infectious disease testing. And then to monitor the adverse reactions, so we have some tests which we conduct there, and some would be totally exploratory for the clinical trial, and so they might be just looking how the adverse event acts or the mechanism of the drug acts in different systems. So that could be some of the flow cytometry, and the molecular testing also might come in that. But like we also have the multi-genomic work into the inclusion exclusion criteria based on the study protocol. So just to all of these core labs which I described are all CAP-CLIA labs, and then we also have the regional approval, with the Ministry of Health from Singapore, like for the Singapore lab, and Ministry of Health in China for the China lab. In addition to the CAP and CLIA accreditation, so we also have the New York State license for the US lab, and then there are ISO certification for our US and EU labs. And we do have just to ensure the quality of those, each QA teams in each of these locations. So, and then there is independent reporting to the, so they report to the corporate office. And then all of them have the global SOPs and the quality manuals and all. And then the training and everything is exactly the same in all of the 4 labs. And we do support all therapeutic areas, including the cell and gene therapies, the infectious disease testing, oncology, and so the variety of trials, in the disease states. And we do have a really good, a state of art of the multi-genomics labs, and with dedicated scientists to validate some of these new tests. And based on the need of the trial, it could be the CAP-CLIA or the exploratory purposes. And so services could be anywhere between the asset development, the nucleic acid, the purifications to the PCR, digital PCR, the Sanger sequencing, next-gen sequencing technologies. And in addition, we also collaborate with big genomic centers, and then to support some of the whole genome and whole exome and the microbiome profiling. And in any given clinical trial, so it's choosing the right platform or the right method or the right test is important. So we do engage with the client, and then suggest some of the methods. But most of the protocols are already written and but we do have some flexibility to suggest some methods based on the protocol need. And so I'm just going to basically go through some of these the validation process. As I said, the time is one of the factor which we encounter. So our validation process there, the actual the protocol which comes to the door is basically through our BD, and then the business development team, and then they pass it on to the commercial team, and then we as a scientific team review the protocol and then extract what testing needs to be done and what can be done within our PPD labs, and if we can't do it, and then we also outsource it. And if any opportunity which is good, makes a business sense to bring that test in, so we identify that test and then we propose that new test validation, and then basically and the cost associated with it. And the moment the client gives the approval, so then immediately our validation team, so they write the plan and order all the reagents and the consumables, resources needed, and all the possible, if they have any difficulty in identifying any positive samples, we usually tend to go with the the spike samples, the oligo synthetic samples. And once the plans are written and then the materials arrive, so the lab takes over and then do the validation data generation. And once the data is generated, so then the validation, it will go back to the validation scientist and then do the the calibrations and the writing the summary, and then go through the approval process from all the lab directors to the quality assurance teams, just to make sure we are meeting all the regulatory requirements. So once the validation summary is approved, so then the actual the service gets offered, and some of the tests we automate the data transfer from instrument to our LIS system, and so the IT gets involved. So just to get to the turnaround time in quickly within 2 months to 3 months, so maximum of 4 months. So all this the validation team, the lab team, QA team and the IT team like work together like really as a well-oiled machine to deliver that validated method. And so like when once like the assay is ready to go, and then they also write the SOP, like the lab takes care of the SOP, and then order any PT material from the CAP, so that we can ensure the quality of the method. So the next phase, like the challenge which we always encounter is some of the regulatory requirements, like from time to time, so they get implemented. So one such requirement is the IVDR regulation, which is been in works for almost like from 2017, and then like in the last year, like May 26th of 2022, so it came into effect. So it's basically like requires any test which is needs the like an inclusion exclusion criteria or the like monitoring for therapy or primary endpoint. So they all need to be either CE mark assay, and if it's not CE mark, at least like you need to approach the government and get the approval for that particular clinical trial. And so for that, like you need to like provide all the validation summary and the need. And then this particular IVDR regulation, like is in the transition phase, like the implementation phase. So I think the whatever the test which we are going to bring it on, like as of like after May 26th of last year, like 2022, has to be like a C approved or like they need to be approved. Like if it becomes a lab developed test, so that lab developed test needs to get approval from the EU government. So that like poses some of the challenges. So even though we might have the technology and but then like by the time you get the approval, like you lose some of the time, and so definitely like you slow down the process of starting the trial, like from the time of the award. Um, so the IVDR like kind of regulation, and I think it's kind of slowed down. It's not loading this slide. Okay, I'll skip that slide. So that basically tells like the IVDR regulation has categorized into 4 categories, like based on the risk. As long as the low impact ones, like it's easy to get the approval, then the high risk ones, like which have the public impact. So then those are like really difficult to get the approval. And I think the molecular testing, most of it like falls under the category 3. That still needs the approval, but it's not like difficult to get. And like within our the organization, so we have like a team called the protocol review team. Like the moment the protocol comes in, and then we identify like which one is the CE mark assay, which are good to go, and then which ones are lab developed tests, so that needs to get validated and get the approval. So that like the identifies and also puts like that information in the budget proposal, so that client is aware when, and then they also kind of take that into consideration whether they want to have the site in that particular region. Like and then the validation team actually identifies the lab developed test and then gives the like make sure like all the validation, all the aspects of it are covered, and then so like all the tests when once it is being done, so we can track it that even LDT assay. And then the lab also like make sure like the results are like in compliance with all of the approved process. And some of the like the experiments, like as I mentioned, if we don't have any the method which is available like in the market, so then we develop ourselves and then we validate. And if it is already FDA approved or CE mark, so we just do the verification process. But if it needs to be like lab developed test, so then we do like more thorough job of doing the accuracy, precision, reproducibility, stability, and establishment of those controls and the matrices. And since it's a global trial, and then like the samples will be coming in from different parts of the world to the nearest lab located, the stability of the sample becomes an important aspect. So, and we could like do only to certain extent, but then just to get the sample from the collection site to the lab, whatever like in the time required. So we definitely validate that part of it before the trial goes on. And then we extend it at then once the trial is started. And then the sample management also is very important in these trials because of the, again, different transits, they're like in different countries, like their customs. And sometimes if it's not properly documented and then if they don't have a good documentation, so they go and sit in the customs and then it will go as a waste, like those precious samples. So we take all of those samples, aspects into consideration when we're moving the sample from one region to the other region. And the spike sample, the controls, that's what we use, like if we don't have the actual positive samples from, from our commercial vendors. And then, but this, material, the validation resources, the AMP, like an Association for Micropathology, has like a really good resource. So if you go there, they definitely give you very good suggestions, what kind of controls or the spike samples one could use it for. And just to give an example here, the method which we validated in our lab, this CMV viral load assay. Initially, we were running the assay for the plasma. In plasma, that's the FDA approved test, using the Roche 6800 platform. And then the client came, they want to also monitor this CMV viral loads in whole blood, urine, and saliva. And then, so since we didn't have the FDA approved test, so we needed to modify this FDA approved test from plasma matrix to different matrices, like in whole blood and urine and saliva. So that's what we did here. And then, as I said, the stability is very important. So we needed to put the urine samples, in the Roche PCR preservative, and then saliva samples, preserved in DNA RNA shield reagent from Zymo. And then the whole blood, EDTA samples are collected, and then so we kind of spiked the CME strain. And then all of these samples again, analyzed using the COVAS 6800 platform, exactly the same as the plasma method. And then we could successfully validate that assay, and then determine sensitivity, specificity, AMR, accuracy, precision, and sample stability. And it, here the sensitivity, in urine is about 274 IU per ml, and then in saliva is about 546 IU per ml, and in whole blood it is about 750 IU per ml. And so the upper limit of all of these, we could do it up to 5 million IU per ml. And the specificity was 100%, the primer probes are very specific. And the and so that's why we could adapt the plasma method to some of these matrices successfully. And as I mentioned, the quality of the method, the quality of the results has to be normalized, in all of our 4 global labs. So in order to get there, so we have the standard process, work processes, the workflow is same, and the instruments are same, and then the calibrators, and then the reagents, whatever we use, exactly the same, and then the SOPs also are the same, so same step. So the only difference is the area, where the location of the lab and the operator is different, but everything else remains same, just to make sure we get the same quality data in all of our 4 labs. So the personnel are also trained exactly the same training program, and then we have the comprehensive management, and then the professional development part. And then we also have another unique the process. Usually when you're the part of the quality control program, CAP sensors the CAP proficiency testing. One day ship about 2 or 3 shipments in a year, and then so we analyze it and submit it, and then they compare with all our peer group, peer lab results, and then give the feedback. And in addition to that, so we also have a program called GLASS. So that is the GLASS, the Global Laboratory Assay Standardization Survey. This program is mainly meant to monitor the quality more frequently. Like, so we do almost like once a month. We analyze for each analyte, like we do the testing in all of our 4 global labs, and then see how the data looks like generated from the 4 labs. So just want to make sure for all the global trial, like all the samples are done exactly, and the data is the same, and then quality is maintained, because ultimately they need to go into the central database, and that data gets submitted to the FDA approval. At that frequency of the QC testing or the PT testing, really helping us to make sure like we are all generating like a good quality data in all of our 4 global labs. So and thank you all. I'm happy to take any questions.